water, forcing the molecule to unfold, and we have our spectroscopic signal. So coming back to protein stability, right? How are we going to get an energy out of this? Well, I told you that it's all about monitoring the population of the folded and the unfolded state. So here again is our simple little equilibrium process. We start folded, we can go to unfolded. We're always in equilibrium. This is an equilibrium situation. Both of these states exist. It just so happens, though, that under normal conditions, the unfolded state exists at a very, very low concentration. We can perturb that equilibrium, again, by the presence of, in this case, again, guanidine hydrochloride. We've also talked about other temperature, etc., etc. So what we'll look at here is now, again, a fraction of that signal change. And this is, again, some data of my students. This is probably CD or fluorescence, I can't remember, of our favorite protein, which is CusF, copper binding protein from E. coli. In any case, what we see is that we start out with one signal in the folded state, but as we increase that guanidine concentration, we flip over to an unfolded state. What this is effectively a measure of, if this two-state model is an accurate representation of the equilibrium process, then what we're really looking at is just a measure of the percent that's folded versus unfolded at any given concentration. So there's our metric of the population. And now, since we have defined the system as being only in one state or the other, that's starting to look like something we could write as an equilibrium constant. And because we can write it as an equilibrium constant, we can calculate a free energy, right? So there's our familiar expression for deriving an energy from an equilibrium constant. And for proteins, we're talking about stabilities in the range of 5 to 15 kilocalories per mole. Remember, our scale of covalent versus non-covalent interactions strengths, right? And we looked at how hydrogen bonds and electrostatic interactions, charge-charge interactions, etc., all have energies on the range of 2 to 5 kilocalories per mole, whereas a covalent bond is at least an order of magnitude more strong than that. Here in this case, we're looking at fairly low stabilities, right? On the order of a couple of hydrogen bonds, really. So what's the story? Well, so proteins are stable, but only marginally so. And what we see here is the fact that a protein's stability, overall stability, is the sum of positive and negative effects. So you have all of these stabilizing interactions that are holding the molecule together, electrostatic effects, hydrogen bonds, etc. And you have a lot of destabilizing effects too. Remember entropy, right? We've gone from an unfolded state that has a very high entropy to a folded state that's very low. So that summation sort of gives you something it's only stable, again, as I said, only worth a couple of hydrogen bonds, really. In any case, we can quantify stability by monitoring these spectral characteristics as a function of solvent conditions. Now, let me say something else about these chaotropic salts. So you maybe have discussed the Hofmeister series in Gen Chem or in organic chemistry. This is just a series of salts that disrupt the structure of water. They alter the structure of water. And in terms of proteins, we can think of the Hofmeister series in the following way. Some chemicals induce unfolding. They destabilize the protein. Urea and guanidine are good examples of this. Other salts stabilize proteins. Phosphate, sulfate, etc. So you could imagine doing a chemical denaturation in the presence of sulfate, and you would be stabilizing the molecule in some way instead of destabilizing. All right. So we can measure stability and we can quantify stability. Oh yeah, well, how do we do that? Well, again, so I think it's useful for us to go through these derivations to see how we actually derive this line. It's one thing to measure the data and you have a lot of spots on a piece of paper, but when we draw this line through it, that's not Excel extrapolating between points. That's us deciding that this model allows us to calculate an energy, and the energy is a value that we want to know so that we can draw comparisons between molecules. So how do we get from spots on a page to the energy? Well, it looks something like this, right? Bottom line, we can relate populations to energies, so we can recast our equilibrium constant in terms of that free energy, and really it's more than just the free energy change. It's also a denaturant dependence to that free energy change. If we look at this curve one more time, we see that there's something that's this isn't just going from unfolded between unfolded and folded. It's doing so in a denaturant dependent way, right? This isn't a step function. It's something that depends in some complicated way on the denaturant concentration. So we build that into our model, that there's two things that determine the equilibrium constant: the free energy of the unfolding process, as well as some denaturant dependence. But leaving that behind for a moment, this allows us to get there. But let's go back to what we're actually measuring. So we're measuring a signal. Well, what's the signal? The signal is a measurement of the fraction that's folded and the fraction that's unfolded. Again, this hinges on the idea that we have a simple two-state system. I'm not telling you about more complicated models. Plenty of these exist, but it's hard enough for us, I think, to get our brain around the two-state version. So what's a fraction? Well, it's the part over the whole. So very simple, right? This is just the fraction of the total that is in the folded state at any given time. And now we've defined our signal as that fraction. Well, let's go back to our equilibrium constant, right? Here it is. We want to do some simplifying rearrangement. That's what I call this, so we can solve for u here, so that we only have a couple of variables in our fraction, and then we substitute, right? So now we're substituting for u in this case, and now what we come up with is, now we're expressing that fraction simply in terms of the equilibrium constant and F, the amount of the protein that's folded. This reduces very easily to 1 over 1 plus K, so effectively we removed that population of the folded and unfolded state from the equation, and now we're simply expressing it in terms of the equilibrium constant, and that we have an expression for, right? So we just plug this expression here, the free energy of unfolding and the denaturant dependence of that change, back into our expression of 1 over 1 plus K, and we are able to use nonlinear regression methods to fit that signal. Remember, this theta is just spots on a page. Now we can fit it according to this model. I want to emphasize, okay, why is Gillespie telling me this? I want to emphasize that in chemistry, we want to write down a mathematical expression that allows us to describe the data. And I think you will agree that this simple model describes the phenomenon quite nicely, in addition to getting us a number out at the end. Okay, so we can derive an expression that allows us to fit the data. Okay, who cares? Well, there's a lot of reasons to care about stability, but something that's easy to get our brains around is simply, for example, thinking about the differences between different kinds of molecules. Let's think about the differences between extremophiles and mesophiles. Let's say Thermatoga maritima is an microorganism that exists at extreme temperatures, right, at the thermal vents in the bottom of the ocean, versus E. coli or Bacillus. Those are more mesophilic. I guess Bacillus could be considered a sort of a quasi or partially thermophilic or thermophilic molecule, and TM, Thermatoga maritima, and a hyperthermophile. So what's the basis of the differences between those organisms? Let's think about it. Thermophile, bug living at high pressure, okay, at the bottom of the ocean, but at very elevated temperatures. Maybe its optimal growth temperature is 90 to 100 degrees centigrade, right? Imagine what happens if E. coli is placed in those growth conditions. Well, all of its proteins denature and the organism dies, right? So somehow they're still using protein, one organism versus the other, the thermophile versus the mesophile. They're still using the same rules for building proteins. So where do we get the difference in the behavior? That's where the characterization of the stability, the quantification of stability comes in. So in this case, we're looking at we're looking at fraction unfolded, but now we're not looking at a chemical denaturation, we're looking at a thermal denaturation. And what we see is, let's see, we're only looking at Bacillus, right, and Thermatoga here. This is a particular protein called Ky. It's a chemotaxis protein involved in bacterial signal transduction. It doesn't really matter. This particular protein from Bacillus in diamonds, we see that it unfolds at about 320 K. ask you what that means in terms of centigrade when we meet in class next week. If we look at TMY itself, the key Y from the thermophile, Thermatoga maritima, we see that there's no signal change. Remember, this really amounts to a spectroscopic signal change over this whole range of temperatures, 300 to 360 Kelvin. We can only begin to see an unfolding transition when we add a little chaotropic salt guanidine hydrochloride. So we add now we repeat our experiment with the thermophile and 1.5 molar guanidine, and we see now that we can see that molecule unfold. But the key here is that the bacillus molecule unfolds in sort of reasonable temperature range, but here we're approaching 100 degrees centigrade, and this molecule has not unfolded. So this sort of experiment allows us to quantify the difference between extremophiles, and we can imagine experiments that we could perform to understand the structural basis of those differences. So here comes the idea that we can use mutation, site-directed or other forms of mutagenesis, to probe these differences, and our spectroscopic handles allow us to quantify those differences so that we can understand the structural basis of stability. Moreover, understanding that the structural basis of protein stability allows us to manipulate systems, and you probably are all aware that lots of biotechnology applications, such as the polymerase chain reaction, require the use of proteins derived from hyperthermophiles in order to function. So we make use of these stability differences in everyday molecular biology. One more slide. Here's the sequence alignment of those two protein. Remember, key Y from Thermatoga maritima aligned with key Y from Bacillus subtilis. Remember, we learned about how to align these sequences last week. So this is just a very simple sequence alignment, and for these molecules it's actually very straightforward. I think there's only 10 or 11 sequence differences between these molecules, so they have a very high sequence similarity, right? And if we look at the crystal structures of these two molecules, you'd be challenged to see any significant difference in their gross three-dimensional organization. Again, this allows us to touch base with our discussion of evolution from last week. However, encoded in those 11 amino acid changes is this radical change in the temperature at which the molecules begin to unfold. We're looking like at least a 50-degree difference, a 50-degree change in terms of the unfolding of the protein, in terms of the temperature that the molecule unfolds. So how is that encoded in this sequence? That's the sort of experiment that we can perform, since we have means of monitoring protein stability. So let's move away from stability for a little while, though, and think instead about the kinetics of protein folding. So in chemistry, we like to think about thermodynamics. That's the stability of the protein, right? We also want to think about kinetics. And understanding the thermodynamics and the kinetics of a system allows us to completely understand the behavior of the system, we hope. So we also turn to kinetics to understand protein folding. What do I mean by that? Well, if you start with an unfolded protein and you allow it to refold, that's a time-dependent event. Therefore, you assign kinetic parameters to that event. So in this case, we're looking at not an equilibrium experiment anymore, but a change in a fluorescence intensity as a function of time in microseconds. Again, this data isn't cited because it's my own experimental work, so you could imagine Blake Gillespie, where did we publish this? I think it was Journal of Molecular Biology, blah, blah, blah. You can go look it up if you're interested. I know you're not. In any case, what we see is that, we begin in the unfolded state, we change the conditions somehow, and I'll describe that in a moment, and then as the system re-equilibrates or comes back to equilibrium, we see that fluorescence change occur over some time scale. Very fast, right? A half-life in this case of maybe 1 microsecond. We'll come back to that idea. So folding and unfolding may be initiated by altering the solution conditions. So in this case, this is a refolding experiment. We've denatured the protein and we've jumped back to conditions that allow folding to take place, and we monitor the conformational change by, in this case, fluorescence. You can do this for both folding and unfolding. It depends on how you do things. You could imagine beginning with your protein in water and then suddenly mixing it into some high concentration of those denaturing salts, and then you'd see again a re-equilibration of the system to whatever the conformation is under those new conditions. In any case, back to energies though, right? So here's a description of a kinetic trace like this. This isn't a very nice single exponential, but as I've written here, but you'll get the idea. We can represent our signal in terms of some amplitude and a rate constant and some time, and then that's an exponential function, which obviously that the data are. Okay, but now we can get back to our energy here by recasting our rate constant, right, in terms of energy. So this is again how we get out our rate constant. What is this activation energy, or E sub A? Well, I draw your attention to this sort of reaction coordinate in which we have a reaction where we go from X to Y, and there is some barrier, right? So EA represents the magnitude of this barrier, and here's a thermodynamic character, that is to say the free energy or the enthalpy change from folding to unfolding. So that's still buried in the process, but the kinetic process is governed by this activation energy that must be overcome. So the higher the activation energy, the slower the reaction. The lower the activation energy barrier, the faster the reaction. This particular diagram was really made with reference to enzyme catalysis, so I'll ask you to store this away in your mind and remember that the way enzymes generally function is by lowering the activation energy barrier. Not relevant for our protein folding discussion, really. All I want to show you is that what this activation energy barrier as represented in the folding kinetics trace, where that comes from. How do we monitor protein folding kinetics? Well, interestingly, protein folding rates vary over an enormous range, microseconds, as I showed you in the last slide, to hours. So you will use a variety of means of accessing those, right? So if your folding rate is very, very slow, right, on the order of tenths of seconds, then you can get away with just manually changing the conditions, right? And that would be squirting in a high concentration of denaturant if you wanted to unfold the protein, or starting with unfolded protein in a high concentration of denaturant and mixing it into some folding permissive condition. But it doesn't take very long before you leave your ability to do a manual mixing experiment behind. You can think about what that would be like, right? You have to imagine yourself holding a cuvette and a pipetter, squirting some stuff into the cuvette and mixing it by hand very thoroughly, and then dropping it in the cuvette. Well, once you get down to a second, a few seconds of folding, then folding rate, then you know that your reaction is going to be over before you even get it into the instrument. So you turn to something called stopped flow, which is simply a pair of syringes that are driven by any number of different methods, but a very common one is just a pneumatic ram, and you get very rapid mixing, very rapid equilibration in the cuvette itself, allowing you to access rates that are more like hundreds to thousands per second. And just to give you a picture of what that looks like, here's a photograph of this is actually two sets of cuvettes, two sets of syringes. Here you see the pneumatic ram that flip a trigger, and the ram forces these syringes together, mixing the folded and unfolded protein together perhaps, and then you can monitor your spectroscopy. There's an optical cell hidden in the back here. So that's what a stopped flow device looks like. But many proteins fold much faster or more rapidly than you can access even by stopped flow. The limit for stopped flow is about 1 microsecond, maybe in some cases a tenth of a microsecond. And as we move beyond those very short time scales, 1 microsecond or so, to even faster time scales, we have to move beyond any kind of mixing period. And there are two methods here on this slide that I'm mentioning for the very fast folding proteins. We use dynamic NMR experiments or temperature jumping experiments. First, let's look at temperature jump. Actually, this is a temperature jump experiment, not a mixing experiment, in which we induce folding by jumping to a higher temperature using using laser excitation. So we have an infrared laser that jumps the temperature of a small volume, and then we, for a very short period of time, we can monitor the fluorescence of tryptophan in that small volume. So that's a temperature jump that's relatively easy to understand. Dynamic NMR experiments are slightly more complicated, remembering that NMR works on the radio frequency time scale, right? So thinking about the time dependence of an NMR response. If the folding process is actually taking place on a time scale that's faster than the NMR time scale, then you will see multiple resonances. Under some certain situations, you can see multiple resonances for a particular nucleus in different conformational states. So instead of seeing one resonance, if it's actually an exchange between multiple conformations, you can sometimes see both of those conformations and extract a folding rate from that. So the bottom line from this slide, however, is that folding rates take place over a very wide dynamic range, large dynamic range, and we employ a variety of different kinds of techniques for assessing those kinetics depending on where we fall in that range. Reminding you that everything that I've been talking about so far is how proteins fold in vitro, when we have purified our protein and we simply monitor the folding of the molecule, that pure protein in solution in isolation. What do we learn when we study those proteins? So I'll simplify everything, boil it down to the simplest terms here. Structures have unique folding reactions. So what we observe is that different proteins behave very, very differently. The context is important for determining how the protein folds up. And in general, though, what we see is that there is some nucleation of structure that happens that determines the route the protein is going to take to fold. Given that, we think that oftentimes protein fold along pathways. And here I say, or do they? Well, there are different situations. For small proteins, we often see folding proceed without the population of any intermediate state. So for proteins that are less than 150 amino acids or so, protein seems to be perfectly two-state. There's only folded and unfolded, and we go directly from one to the other. How is still a question, and how we address it in the absence of intermediates is also an interesting question. But we also see for larger proteins that well-defined intermediates are populated during a folding kinetics experiment. So this gives rise to a couple of questions that are relevant to any kind of kinetics analysis. What's an intermediate? What's the definition of an intermediate? Well, let's just say that it's a species that's populated according to whatever method we're using, right? So whatever technique we're using to analyze the kinetics, we can actually see the population of a third or fourth or fifth state, right? Not just folded and unfolded. So they're measurable populations. What's a transition state? Well, normally we used to thinking of a transition state as a very unstable structure, right? And that isn't populated to any great extent. So it's a little bit challenging to think about what the structure of a transition state is. But we'll just come to this discussion again when we talk about folding, not folding, but enzyme mechanisms and the importance of the transition state there. Suffice it to say today that these two ideas, identification of folding intermediates and identification of structures that are important for the transition state of protein folding reactions, are both important to understanding folding mechanisms. Thinking a little bit harder about mechanisms,